Hi everyone, this is Pedro Murua, and today I'm going to show you the preliminary results that we have so far in our GSS research fund called Algal Blister. To put you a little bit in context, this project was designed essentially trying to look for some weaknesses related with algal diseases in the region, right? We are currently collaborating with 4 different countries beyond Chile, 5, sorry, which are Argentina, Peru, Brazil, Mexico, and Colombia. And with these countries, we essentially are trying to push the discovery of new threats in terms of algal diseases in the region. But before that, when we were essentially designing this project, we noticed that we got 3 different main gaps in the country, in the region, that actually are preventing us to potentially develop a sustainable aquaculture in the future. The first of it is related with the lack of capacity building or infrastructure, specifically dealing with problems of algal diseases. This is, for example, discovery of new diseases or diagnosis of already known diseases. And this is, in this sense, the first objective that we were trying to deal was trying to create capacity and infrastructure in the region to tackle these problems. The second one was mostly related with the state of the art about algal diseases that we were not aware. In that case, the second objective then was related to the generation of a first baseline of the currently known diseases in the region. And the third one is most related with the policy, and specifically, we were not aware how much our biosecurity related policies were covering our backs when we were developing aquaculture activities. In this case, the third objective then was trying to revisit this literature, generate a baseline, a database with all the policies that are somehow related with seaweed aquaculture and how much they are covering aspect of biosecurity risk in their lines. So in relation with the first objective, we are happy to say that we were able to develop a seaweed and phycopathology lab in Chile, essentially. This lab was developed in Universidad Austral de Chile, UACH, with an input coming from both GlobalSeaweedSTAR Algal Blister project plus Universidad Austral internal funds that were able to complement all the equipment that we were not able to acquire by Algal Blister itself, right? At this moment, with these 2 big inputs, we are now able to not only diagnose some diseases or basically study new diseases coming up from for example, seaweed aquaculture, but also we are able to develop, to pilot some specific experiments that are going to help us to, for example, study the prevalence of a disease in terms of amount. So we can do some epidemiology, and also to develop some tools to understand stress in seaweed, for example, measuring PAM fluorometry in the host, right? With all of this, now we are currently trying to apply to different proposals in collaboration with the collaborators from Latin America that I mentioned before. And actually, we already got one project granted to be developed in Antarctica, in the Chilean base located in Antarctica. Now with the lab facility in place, then we wanted to develop a tool that can help us to gather more people that can send us potential disease samples, right? And for that, we basically collaborate with SAMS. SAMS currently has a webpage that you maybe are aware that is called My Seaweed Looks Weird, where phycophiles can send some samples, and later on this information is gathered by some scientists. They receive samples as well, and with this they can offer a in-kind diagnosis for such diseased seaweeds. In our case, what we did in collaboration with some people was to actually transform a little bit this platform into a Spanish or Portuguese version. So essentially farmers that are not very familiar with English can submit samples here in Latin America, and these samples are then directed to me in Chile. So essentially by this way, we are receiving and treating or generating a diagnosis timely and generated locally, which is also very important for people around here. Then we were working generating a baseline of algal diseases. This was done initially gathering all the literature that was mentioning diseases of some sort. And in this case, we were able to compile the literature coming from the last 80 years. And from them, we noticed 33 scientific articles mentioning 130 different records of algal diseases. Yeah, and these reports are then plotted here in this map, you can see. They are very variable. The most newest reports are pretty much viruses or pseudofungi as well. But the older one are related with bacteria, for example, or fungi or algal endophytes. Yeah. We can also notice a very disbalanced reporting material, where most of it is coming from Chile and Argentina, some point Peru as well. But as you can see, most of the Caribbean area, for some reason, is not really documented, fortunately. So with this, we started to notice, because if you start to picture the whole world and the whole world diagnosis effort, you can notice that here in Latin America, including Chile and Argentina, for example, is very, very underdocumented, and it needs dramatically more effort to actually gather, grasp all this biodiversity that we are missing. And to make a contrast, for example, in the framework of this very project, we were doing some sampling, some sampling effort. You have to consider that we were doing this during pandemia, which meant that we were not able to sample as much as we really wanted. Yeah, but to make you more or less clear, we were doing something like 6 different sampling in Chile and 1 in Peru in 6 months. And during this sampling, we were able to note 22 different records from casual sampling. And this is not complete. I will say that we probably have at least 10 more that are gathering now. And with this, I don't want to highlight our very capacity to make a diagnosis fast, but instead to make it clear that we really have a lot of work to do in the region. Yeah, and here I try to shoot you with a little bit of this record. Most of them, I have to say, are coming from cordillerian endophyte, brown algal endophyte, growing inside red or brown seaweed. We also notice some pseudofungi like omycete or phytomyxete infecting southern ball kelp. In some cases, we were really doing some work in kelp gametophytes that are here available in our own germoplasm, and that are actually being used for kelp aquaculture in the country, in Chile in this case. And here we notice that at least we have 1 confirmed molecularly present in our strains, but also we have at least 10 candidates that are now being sequenced so far. So yes, in the end, what we want to say is that we are just with this project and with pandemic context able to provide something like 20% or 50% of the total records in 80 years, which is quite a lot, and which meant that we're missing a very big picture in India. In this project was also very, very difficult to overlook what was going on with the so far the only commercial culture that is going on in the region, that is happening here in Chile, and that one is the culture of Gracilaria, or ex-Gracilaria, now it's called Agarophyton. where in this case, we were able to also make a baseline of pest, not disease itself or not true diseases, but still very nasty for the industry. And after making this database of different pest reports, we noticed that at least we have almost 500 reports in the country. Yeah, and if you can see the maps, both of them, you will notice that they are very nested to 3 different areas that are the ones where this seaweed is being cultivated the most. And at least the 95% of these reports are coming from the south, which is where roughly the 80% of the biomass that is produced via aquaculture is produced. What you can find a lot of things, and to be honest, we don't know yet with real clarity what it is, because most of these species are characterized mostly at genus level. But if I have to sum up them, we have a very important amount of ceramelian, etocarpelian, or even green tide that are being common which are very common lately. And in some cases, we also have some reports of diatomom fells as in the euchemoid farms. So what is Alga Blister doing for this now is, well, we were able to get some strains that are not being isolated, but most importantly, we're trying to characterize them, in terms of, using DNA barcoding to essentially get a more accurate taxonomic affiliation for all of them. And then is we got the third objective, which is related with the biosecurity. And in this case, I'm not gonna go into the detail in this, but we have completed at least 2 different countries, Chile, that we are using as a study case and to polish the protocols, and then Peru, which is one of the countries that we were aiming to look into. And in this case, we are indeed able to build this database with all the policies that are related with seaweed aquaculture and how much of them are actually mentioning a little or something related with biosecurity, risk prevention, or mitigation. And in this case, I have to say that we have to develop different approaches to basically interpret these legislations. And in general, in the approaches that you can see above, we call them relaxed because they consisted in noticing at least a mention, even if it was superficial, about the dealing or the prevention of certain biosecurity risk. Yeah, and in that case, effectively, we have a significant body of legislation that are trying to deal with this, but it's only mentioning in a very light way. Because when you start to look into the detail and to notice how this legislation or this part of the legislation is actually dealing with a biosecurity risk, in this case I put only the cases for ecological and environmental, you can see that this specificity is actually lost, right? And with this, what I'm trying to say is that, okay, there is an intention from the legislation to actually cover up a biosecurity risk, but this is not in the practice carried out by the same frameworks in both countries, right? Which means that we have a very big gap between what we are essentially offering to the citizens, to the community about avoiding this risk, and what we're actually doing to preventing from that. And lastly, related with most with capacity building, and as you may know, we were unable to gather presentially, so we had to join forces with the Chilean Phycological Society, where essentially what we are doing now is organizing 2 different events in the framework of this conference related specifically with algal diseases. The first of them is a plenary given by Claire Gachon, a member of GSS-STAR, and also a very big person related with algal diseases, which is going to show us what is her lab doing in these terms. And also we are organizing an algal disease symposium that is majorly focused on showing different approaches, experimental approaches, specifically with different lab models or field model that are using a pathosystem as a center of the research, right? And in this case, is myself plus 3 different young researchers coming from Canada, coming from Germany, and New Zealand that are gonna talk to us. And when I mention to us, I'm talking about my collaborators from Latin America, about different approaches that these collaborators can do in, for example, their labs when they are studying, I don't know, viruses or bacteria or red algal parasite, for example. So to sum up the impact that we are foreseeing from our project, I could say that may sound trivial, but we are the first and hopefully not the last or not the only lab that is now trying to help creating consciousness about the problems that can be generated by algal diseases, for example, in aquaculture. And we are not only trying to generate this consciousness, but also trying to help with farmers, for example, to tackle these problems locally. The first thing that I will also like to mention is related with that we are finally knowing our algal biodiversity. We're far away from what we should know at this point. But more remarkably, I would say that we are knowing that there is some beast around that may eventually affect our cultures or affect the seaweed that we are harvesting from the wild. And the first, and the third thing is that we are also being aware that our biosecurity may be not as good as I will have expected. And this is a message that we can send to our citizens and also to our policymakers in order to essentially, and I will say this is one of the most important things, to make changes that may at some point ensure this very incipient seaweed agriculture that we have in the region to flourish properly and sustainably. So this was me. Thank you very much. I would like to acknowledge many people. I'm not going to name them all, but with special emphasis with my 4 collaborators from different countries in Latin America, Danilo, Cecilia, Leila, and Daniel, and also to the stellar team coming from GSS that have been helping me either with different things related with the project or related with SAM itself. Yeah, remembering that I have this double affiliation. So thank you very much, guys, and well, hope you enjoy these preliminary results. Bye-bye.